so that is 4:05, which means it's a good time to start. So, hello everyone, welcome to today's NIRx webinar. Today we're going to have a talk about multimodal integration of fMRI and fNIRS signals from protocol development to data preprocessing. We have our invited speaker here with us, Dr. Sara Sanchez-Alonso, who is currently an associate research scientist at Yale, but I'm going to give her a better introduction. If you don't know me or my colleague Cami, I'm going to skip a few slides to talk about us, and then I'm going to come back. So the two people that you see on your screen are me, Giovanni, but everyone just calls me Gio. I'm the support lead here at NIRx. So if you've written a support ticket, you've probably seen me before. And Cami is our marketing and event coordinator, is the one making all of this happen behind the scenes. So let's all thank Cami for this webinar. And who are we? We are NIRx. So going back to the first slide, if you're not familiar with NIRx yet, we pride ourselves as a solution provider in neuroscience for applications of fNIRS. And we call ourselves solutions provider because it's not like we give you a box and then you're off on your own. We establish lasting partnerships with labs and scientists from all over the globe for over 20 years now. If you'd like to hear more about us or talk to us, you have all of our socials here. This is more on like a talk to us kind of way, but if you want to talk to us more on a consulting level, you can email us at consulting@NIRx, or if you have a support question, you can ask us a support question at support@nirx.net. We do have a newsletter. We send 4 emails a year, so no spam on your inbox. So if you want to keep up to date with what are we up to, what's the next fNIRS device, what's the next development in the field, or is there some workshop happening, some training happening in your area, feel free to subscribe to our newsletter. As I said, we will not spam you. Yeah, considerations of the webinar. You are all muted, but questions are very welcome, and we would love to answer them. Please use the chat to ask your questions. This webinar is recorded and will be available publicly for free on the upcoming days. And as I said before, if you have questions for your own experiments, or if you want to achieve some of the very, very awesome science that Sara is going to show to us today, you can contact us and we can talk about it. Okay, so without further ado, I would like to introduce our speaker for today, who's here to talk about a very exciting multimodal application, as we have all known from the title of the webinar, combining fNIRS and fMRI. As I'm sure most of you know, this is a big showcase of one of the awesome capabilities of fNIRS being so friendly to other modalities and allowing you to perform very high impact science. So I'm sure Sara will showcase it much better. So without further stalling, Sara is here today. She's an associate research at Yale. Her primary focus is on human language and particularly interested in early childhood. Her work combines fMRI and fNIRS to map developmental neurobehavioral variations, and with the general aim to inform typical and atypical development and advance paradigms for cross-age comparisons of language acquisition. So a full plate from methods to applications. So Sara, I won't take the stage for any longer.
Thank you, Gio. Let me see. I'm gonna share my screen. Yeah. All right. Can you see my slides?
Yes.
I can. Yeah. Okay. Perfect. All right. All right. So, all right. Well, thank you. Thank you everyone for being here, and also to NIRx for the opportunity to talk about our work, which it's been in the works for about 3 years now. And this is where I'm gonna talk today is multimodal integration of simultaneously collected fMRI and fNIRS signals. I want this webinar to be practical in that, you know, we've troubleshoot a lot in the last 3 years. So I want it to be useful for those who are thinking of doing a similar study, so they don't have to go through the different kinds of troubleshooting that we had to do. So first of all, I'm gonna begin by saying that this is very much a team effort, as you will see. Not a single person could not have done this study. Die Kaslin and Arana Egerbrecht are the senior authors in the study, but there is a whole team behind that has been working together for a few years already. And I also want to acknowledge the funding sources, and of course, the support from NIRx, who's been, very, very supportive throughout the whole process. So let's begin. And first, oops, okay, yeah. So first, I want to start by thinking about why do we want to collect simultaneous fMRI and fNIRS signals, right? I think that any multimodal imaging study needs to first answer this question. It requires a lot of effort and time. And I'm going to give you some examples of why we want to do this, right? But this is just kind of like so that we can brainstorm together and really think whether, this is really something that you'd like to do. So you may want to validate the fNIRS signals against fMRI, which is the gold standard methodology in neuroimaging, right? So that's something that may be useful for your study. It could be that you're interested in testing a given experimental paradigm to ensure that you're covering the regions of interest that you care about outside when using NIRs outside the scanner. You may be interested in further assessing the interaction of cortical and subcortical regions, right? And then you have the mapping between the two signals that is going to help you when you collect data outside of the scanner. Or maybe you are using fNIRS with developmental populations, for example, or clinical populations, and you want to establish a comparison with, for example, an adult sample, right? You want to collect fMRI and fNIRS in the adult sample, and then do only fNIRS with a developmental population, right? So these are some of the scenarios that in which this kind of multimodal imaging approach may be useful, but of course there may be others. And so before I move on to the practical side of the webinar, I want to start by thinking about where do we come in, right? If you've read the fMRI&fNIRS literature, you'll probably notice that there have been many studies before us who've done this kind of multimodal imaging approach. And the first studies were the foundational studies in the field happened in the late 90s, early 2000s by Kleinsmith et al. And Toronov et al., looking at the spatial specificity of the fNIRS signal during a finger tapping task, and looking at the spatial and temporal correspondences between the both fMRI and fNIRS signals. And at that time, they already observed that indeed there are correspondences as expected. Another study that came right after by Strandman et al. Showed that there are indeed strong correlations between ball and hemoglobin species, again using a finger tapping task. However, the correlation with the oxygenated hemoglobin was the weakest. This is probably not surprising now, given what we know. And they also show highly variable correlations across subjects. So after these foundational studies, I want to call your attention to kind of like a shift that happens around the year 2010, right? I'm skipping, of course, several important studies in the field that happened before 2010, but in the interest of time, what I want to call your attention to is this kind of shift that happened around 2010, 2011, in which the studies started to go beyond motor task and start exploring other type of experimental paradigms. So Queero, for example, show that NIR signals are highly correlated with fMRI, but NIR signals have weaker SNR, particularly if we look at brain regions more farther away from the scalp. They also used different paradigms, visual and motor, but also higher order cognitive function paradigms. And in societal in 2012, as another example, they started to look at resting state networks and noticing that the same networks that can be captured by fMRI can be observed in the fNIRS signal. And there have been other studies since then, right, that I'm going to skip in the interest of time. But basically where we came in is trying to address some of the limitations of prior work. So prior studies had been limited to a specific ROIs, a maximum of 24 channels. They didn't have short channel measurements just simply because they were not available. Physiological signal regression was not assessed. And there was no assessment of test-by-test reliability of the signal, which has been a very important topic in the fMRI literature. So those are some of the gaps that we wanted to address with our study. And now I'm going to move on now to the actual approach that we took and the multimodal imaging component. So there are going to be two parts in my talk. First, I'm going to talk about the protocol that we developed for collecting the data. And then I'm going to talk about the analytic pipelines for preprocessing fMRI and fNIRS signals and some of the aspects that are important to keep in mind when preprocessing the two signals. So let's begin with the experimental protocol first. What I'm going to present here today can also be found in detail in a publication that was published in the Journal of Visualized Experiments last year. So if you want to go to that publication and look at in more detail so some of the details of the protocol, you're going to find in there. So for fNIRS data collection, then we used a NIRS Scout X. Some of you may be familiar with this system. So here's an image of the system. Of course, in our case, the optical fibers were much longer, right? As you can see in this picture, they were 10 meters long. So these are the fibers in the control room. And then what we did is that we moved them through the waveguides into the fMRI room. We built this bridge. It's made of plastic. That allow us to transport the fibers to the MRI scanner. And if you're thinking of doing something similar, it's very important to keep in mind to take measurements along the way before you get the fibers, of course, and then the distance between, in our case, the drawer and the scanner, right? That's, of course, it's going to be very important. With respect to NIRS cup preparation, we had to implement a few things in addition to what NIRx already provided. So if you haven't seen the cups for fMRI NIRS data collection, this is an example of the cup. As you can see, one of the key differences with the traditional optodes that you can see, for example, in the NIRS port or outside of the scanner is that they are flat, right? So they allow the participant to lie down without problem, given the flat surface. Something that we did in order, and this is going to be relevant later on when I talk about co-registration, is that we sew vitamin E capsules next to each optode on the NIRS cap. So we have 4 different sizes, and this was obviously a very time-consuming process. But if you really want to make sure that if you really care about the location of the optodes, and in our case, we did because we wanted to validate the fNIRS signals against fMRI. I definitely recommend it. It's time consuming, but we just had to do it once, and they stay in the same location throughout the whole study. As you'll see later, we've collected data from 40 subjects already. So I'd be happy to talk more about how we did this. If you're interested in the type of material, it's also fun. You can also be found in the publication that I mentioned earlier. This is how the cup looks. As you can see, what we do, we have a whole head coverage, right? So what we do, and this has worked very well for us, is to bundle the fibers into these 4 different bundles. So we have these cable organizers that are made of plastic. And we just bundle them into 4 different bundles. And that allows the cables to stay organized, and it doesn't interfere with the participant while they are lying down. So I recommend that kind of arrangement if you are doing a full head coverage. With respect to head coil preparation, so we had to use the 20-channel head coil because the 32 and 64 channel head coils were too tight. So we wouldn't be able to fit an adult head. And we spent a lot of time thinking about this. it was probably about two months that we started to troubleshoot this about, in terms of comfort. Like we were really concerned about comfort, about the quality of the signal. So we did a lot of testing. We used different type of pillows, different arrangements, and in the end, it worked out really well. And after testing 40 subjects, only 2 people had to leave the scanner before the end of the session. And I think that really had to do more with their sensitivity to the optos more than anything. But this worked out really well for us. And basically what we did is that we are using the pillow that comes with the Siemens scanner. And then we have this little phone that we allow the participant to adjust based on comfort, right? So they adjust it at the beginning of the study and they leave it there for the entire duration of the study. And the study for us lasts 40 minutes in total, including structural scans. I'll go in detail in just a moment. This is how it looks. So the participant is resting on that pillow. As you can see, there are the cable organizers ensuring that the participant is comfortable. With respect to the montage that we use, so we decided to go with a full head coverage because of the type of experimental paradigms that we were using. We wanted to make sure we had access to the full head. And you can see that we also use short channel measurements provided by NIRx as well. So you can see the green circles is where the channels are located. So we have 8 of them in total, and we opted for a distributed for a distributed arrangement of the short channels. And you can see here a picture of the short channels. I think that there is a newer version. So if you get them now, they're probably going to look slightly different. It's an improved version. And as you see, I think in my next slide, yeah. We also have these rubber buffers. So these rubber buffers are really important for areas that don't have hair. We only use them for the frontal areas. Apart from that, we didn't use them at all. That we did have to test them in advance. The rubber buffers that we got at the beginning that were black, they actually had metal inside, pieces of metal inside, and created strong artifacts in the scanner. So I would recommend to test them in advance before you bring a subject into the scanner. Here you can see some of the preparation tools that we use for the cups. So you can see the cable organizers. We use the ear picks for hair removal. So these ear picks, what we do, we remove the metal inside them. So they come with a battery if you want to light them up. So we had to remove that, of course. And then we had tweezers. Those were also provided by NIRx to remove the optodes. So if the calibration wasn't as expected and we had to remove hair, that's what we would use for optode to remove the optode. Something also that we did that worked out very well for us is that the grommets, we actually glue them to the cap so that they wouldn't move. So that allowed the opto to basically not wobble at all and be very stable. Yeah. Another important step in multimodal imaging, and in particular for NIRS and MRI, is signal synchronization. So here you can see the trigger setup. This is the parallel port replicator that is provided by NIRx as well. It's quite straightforward. And what we did is basically we were getting triggers from the scanner. So in every TR we would get a trigger, and we would synchronize the experimental script and the NIR software to the scanner, and that worked out really well. So I strongly recommend this option. Next, for fMRI data collection, we had a Siemens 3D Prisma scanner. Again, we used a 20-channel head coil, and that's the one that worked best for us. We also collected simultaneous data physiological measures. So in particular, we collected heart rate and respiration rate because we wanted to use them for regression of the signals. For skeletal anatomical registration of the fNIRS signals, we use a couple of methods. So as I mentioned earlier, we have vitamin E capsules sewn next to each optode on the NIRS cap. So you can see them here on the T1. They are very easy to see on the T1. I would recommend looking into smaller vitamin E capsules that are small. There is a wide range, and the smaller is usually the better. That's what we found. And we also used a 3D structure sensor. This is just simply because we wanted to look at the comparison of the two. The 3D structure sensor is something that we use outside of the scanner with developmental populations. So we wanted to see how co-registration compares when we use vitamin E capsules versus the 3D structure sensor. And this is something that we are still working on. If you're not familiar with the structured light 3D scanner, this is provided by Occipital. It's a scanner that projects a known pattern. In this case, it's going to be the head onto a scene. And the way this pattern deforms when it's striking the surface, it allows the scanner to calculate the depth and surface information of the object. We are also pretty happy with it so far when using it for adults. If you're testing toddlers or younger kids, it can get a little tricky because it's sensitive to motion. But so far, for adults, this is what we've been using. And you can see that you basically go around the subject, capturing the head. It takes about, I'd say, about a minute or maybe even less. So now we're going to do a recap of the experimental protocol so far. So what I've shown so far is our data collection device, the NIRx Scout X. We talked about NIRs cap and head coil preparation. I've also shown you the full head montage that we use and then the short distance detector setup. As I mentioned, we are collecting also physiological signals, and importantly, we have a trigger setup for signal synchronization. We are using two different co-registration methods for skeletal anatomy mapping. Okay, so I'm going to move on now to the next part of my talk, in which I'm going to focus now on the analytic pipelines and some aspects that are important to keep in mind when comparing the two signals. So first of all, as I mentioned earlier, the type of experimental paradigms that we're using it was important for us to use a full head montage because we have a variety of paradigms and we wanted to make sure we had access to the full head. So we've collected data from 40 young adults, and they come for two sessions. In the first session, they do two movie watching runs for a total of 15 minutes, and they do two resting state runs for a total of 10 minutes. And these are data in which we are analyzing the functional connectivity profile and how it compares to fMRI. And they come for a second session, and in the second session, what they do is again they do another resting state, two resting state runs, because we were interested in assessing the test-retest reliability of the signal. They also do a story math contrast task. It's basically a language task in order to map language regions for about 10 minutes, and then they also do a flashing checkerboard run for about 6 minutes. And today I'm just going to focus on the flashing checkerboard task, and happy to talk about the other task as well if you're interested. But for in interest of time, I focus on the visual task. So first of all, let's talk a little bit about the data workflow, and I'm going to point you to some key things in this graph. So first of all, the software that we used is we use Neurodot for processing of the fNIRS data, and we use QNX for processing of the fMRI data. Something to keep in mind when when comparing the two signals is that you're going to need the structural images if you want to have a precise mapping at the individual level of optode and anatomical location, right? So what we did is we collected both T1 and T2 images in every session, and then that allow us to do image reconstruction of the fNIRS data. So if possible, if you can collect both T1 and T2, that would be ideal. Another thing that we were interested in doing is to assess the sensitivity profile of our NIRs array. So I'm going to talk a little bit about that in just a moment. And finally, once the two signals are mapped to the same atlas or to the cortical surface, that's when you start the comparison, right? That's the subsequent steps are going to be the same for both methodologies. So let me show you a little bit about the sensitivity. Let me talk to you a little bit about the sensitivity profile. So what we did is that we calculated the sensitivity profile of the array using the head model to assess the field of view. And what we did is that this allow us to get an idea of where can we capture signal from, basically, with our montage. Our montage, as you can see, is sparse, so it doesn't have overlapping channels, right? So it was important for us to kind of have an idea of how, where are we getting the signal from? And then this model sensitivity for each measurement is especially registered to an atlas space, as I mentioned earlier. Um, Next, in order to, I'm going to show you a little bit of the quality assessment that we did with the raw data. I'm going to show you individual level data. Later on, I'll show you a couple of their subjects. But whenever, if we've observed any variation across subjects, I'll let you know. So these are the mean light levels for each of the optoids in our cup, and what coupling is going to be shown in white and yellow, as you can see here. So we're always going to see worse coupling at the top part, so basically close to motor areas. This is an example of one of the subjects. The quality of the data varies across subject, but it's usually pretty good. Just the cup setup is the time-consuming process. Actually, it takes about 10 to 15 minutes, but really allow us to ensure that we were getting good quality data. Of course, these are all adults, and there is minimal motion during the scanner. We also look at signal to noise ratio for cardiac pulse frequencies. And again, you can see here that the SNR gets worse close to motor areas as expected. We had some issues in some subjects around the occipital areas, primarily because of hair. It wasn't always the case, but that's something to keep in mind if you're really interested in doing in looking at visual areas, right? That's going to be an area that you have to be very careful, making sure that you remove most of the hair. We also look at the temporal variance of the data. So here what I'm showing is a histogram of the percent signal variation. And basically what we did is that any measurement that is above the threshold of 7.5 is considered a good measurement, right? So you can see that most of the data is good. There are some, of course, this is a very good subject, but it's not an exception. I would say that the quality of the data is pretty good. Of course, these are adult subjects, and here you can see the good measurements. In this participant, there was only one channel that we lost. In some of the participants, again, as I mentioned, occipital areas were the worst, just because of hair, primarily. Temporal areas, we were fine, and frontal as well. I'm looking at now, I'm going to move on to look at data quality in the pre-processed data. So basically what we did is that we look at the data after filtering low and high frequency noise. So we did this for every subject. And then we also did a Fourier transformation of the data just to make sure that we could really see the peak at the stimulus frequency, right? So, and this is something that, very reliably is identifying, the subject that are going to have good data. So the validation of, I'm going to now show you the results of the flashing checkerboard task for a few subjects. And here, just to tell you a little bit more about the paradigm. So what we did is that the subject was exposed to about 10 seconds of a checkerboard, and there was an intertrial period of for about 20 seconds. And it went on, so on and so forth, so in periods of 10, 20, 10, 20, on and off for about 6 minutes. And what we can see is that at the subject level, this is the block average data of the NIRs, of the block average NIRs data, and we can see that we see very clear bilateral activations in both left and right hemispheres. This is the case for pretty much every subject if we don't, in cases in which we don't see one of the hemispheres, it's usually because it was blocked by hair and it's easy to identify. Here we can see something that we also did is to look at the visual cortex during the flash injectable period, right? So making sure that in every stimulation, we actually get this increase in oxygenation. And we also look at the same data at the same point between flash injectable periods, ensuring that we see the deoxygenation levels going down. And if we look at an unrelated brain region, we see that we don't see these peaks at the stimulation frequency, at the stimulation period. So we did this for every subject, making sure that we are actually getting the data that we expect in the visual cortex. And here is an example of about three subjects. So we can see that there is variation in the peak of the hemodynamic signal, and there is actually substantial variation across subjects, but we always see the peak and then going down, right, for the block. And here are the subject level results for the fMRI data. So here, these are the same three subjects that I've shown before. And here what we were looking for is really making sure that we were able to capture the visual V1 and V2 with the fMRI data as well. So there wouldn't be any artifacts. We were, at first concerned whether the opto pressure would create any artifacts, for example, but that's not something that we saw. And we actually, at the subcortical level, we also see the LDN activations. And again, we see it for fMRI, we see a variation across subjects as well. And there's an upcoming preprint, so stay tuned for that. In this preprint, what we are going to see is an fMRI-based validation of fNIRs. We are with the flash injectable task. So what we are looking at here is what we are going to be showing in this manuscript is the time series temporal correlation for the fMRI and fNIRS signals and how this correlation varies according to cortical regions, but also location of the probes, right? So what happens if the probe is farther away or closer to the activation, and also looking at what happens when the activation is closer to the cortex or farther away from the cortex. Something that we're also looking into is what happens if we change, for example, the smoothing parameters when analyzing the fMRI data. So stay tuned for that. We're also doing a comparison of SNR across the two signals and looking at the topographical similarity of fMRI and fNIRS signals. There is also, given that we have 40 subjects, something that we're also interested in doing is looking at the individual variation across subjects and really seeing how these different comparisons vary across subjects. And I finished earlier than I had expected, so that leaves us quite a few some time for questions. So thank you everyone for your attention. I'll be happy to take any questions.
Thank you so much, Sara. This was awesome and even shorter than expected. So, as I said before, during the introduction, we will take questions in the chat. But if you want to ask your question right now, feel free to paste it in the chat, and then I'll call you. I see one question.
Just very quickly, your audio is coming in and off a little bit. So I can hear Gio because he's at the office, and Sara, he was saying like this webinar was awesome, and he was giving you all the positive feedback, and then we didn't hear any of that.
So, can you hear me better now? I will give you the first question while I reset my headphones. So the first question is the one in the chat room from James from where the vitamin E capsules placed on each source of detector in one.
That is correct. Yes. They were placed in each source and detector. And we did that because we wanted to, when identifying the channels, we wanted to have, what we needed was the coordinate for each source and detector, right? And then what we are doing during the co-registration process is identifying the channel and then mapping that onto the underlying brain anatomy. Yeah. Should I answer the next question, Jill? Yeah, okay. Yeah, so the purpose of the vitamin E capsules is to make sure that we can reliably identify the location of each channel, of each fNIRS channels with respect to the underlying anatomical region, right? Because what we want to do is do a validation of the fNIRS signal with fMRI, right? So in order to do that, we really need to know where are we capturing the data, the fNIRS data, and then do that comparison with that given voxel or region of interest with fMRI. So that way is important, right? Something that we consider at the beginning is, do we need vitamin E capsules for every optot? And for us, it was worth the effort. It takes some time, but then you know exactly where each optot is located, and you just stay for the full study. So I would recommend it. Is the data going to be publicly available? Yes, we will make it publicly available. Yes, for sure. Yeah.
Okay, I should be better now in terms of audio.
Mm-hmm.
Great.
Did you
Good.
use the structure sensor before or after optot placement in the fMRI room? That is a great question, and we actually thought about that for quite a while. We ended up opting for doing it outside of the scanner. So basically, we put the cap on, and we do it outside of the scanner before the optot placement, right? And then we bring the subject in. Of course, there is probably, as we are capping the participant, that is going to probably, lead to some movement in the cap, right? So that's something that there is nothing that we could do about that, right? We try to be as careful as possible, right? But we just couldn't go around the subjects while they were wearing the cap with the optots and take the scan with the 3D structure scanner.
Okay, awesome. So as long as we don't have questions from the chat room, I will move to questions that were asked during the registration process. So trying to filter for things that you already covered during the webinar. Are there any additional considerations that are taken to ensure participant comfort during simultaneous fMRI and fNIRS? You did mention it a little bit during the presentation, but if there's any additional ones.
Yeah, for sure. So apart from the cap, which is what we were concerned about, and actually worked out pretty well. Something that we noticed is really there's a little bit of variation with respect to participant sensitivity, and the smaller the head, the more uncomfortable it gets, right? Because if you're using a 54 versus a 60, right? In the 54, the optos are going to be closer to the grommets are going to be closer together, right? So that's going to be a little bit more uncomfortable for the participant. So we noticed that in general, 56, 58, 60, they were fine. People who were in a 54, they were more sensitive. So we didn't actually have that many people with a 54 centimeter head, but the ones that we had at some point, we were aware that this was going to happen. Yeah. So that was it in terms of comfort. At first, something that we had thought, because we were not, I had been inside the scanner myself with the cap, right? And it was 40 minutes. It was like, okay, I felt like I could do it, but we were thinking, what if participants cannot do it and we have to stop in the middle of the session, right? So we were considering that option if participants would ask for it, but not people in the end didn't ask it. So I think there was just one participant that was claustrophobic and had to stop. That's it. Oh, and another thing important that I'd like to mention actually with respect to comfort is the placement of the headphones. So we use the noise canceling headphones that are provided by Siemens. Anybody who's collecting fMRI data are familiar with them. They're kind of flat, right? So what we did is that we would position the participant in the scanner, then slide the optodes very carefully so that we wouldn't move the grommets and the optodes. And then we would put inflatable pillows if needed to make sure that they would stay on, right? That worked out well. we had to, there was a little trial and error until we got it right. But once we got it right, I think by the 5th participant, it worked out well. Yeah. So that's the other thing that I would mention. But apart from that, we were actually very surprised how, that so many people for 40 minutes, they were able to do it without problem.
Awesome. So I have another question here more towards the methods or the analysis. You mentioned using MRI to validate fNIRs. Reversing the question, how do you value fNIRs informing fMRI, if any?
Right. So I think the fNIRS, the advantage of fNIRS is that you can use it outside of the scanner, right? So you don't have the constraints of the environment, right? So it's portable, it's wireless, right? And I think the way that fNIRS can inform in that is that, all we know about the brain pretty much is because it's through fMRI, right? But what happens when we interact in the real world with people? We don't have that data, right? And I think that is a gap that fNIRS should be filling, right? Like what happens in the real world, what happens in naturalistic environments, right? And so I think fNIRS has a big role to play, right? Multimodal imaging studies can help bridge kind of like this gap between the fMRI field and the fNIRS field, and really show that the same type of data, if you're doing a very careful experimental design and pre-processing, it can be used as a comparison, right? And then I think that the advantages are huge, right, in terms of what fNIRS can do. Yeah.
Thank you very much once again. Just looking at the registration questions again. So multimodality of fNIRS, fMRI, and potential artifacts, if you have any comments on that.
So this is something that we thought about at the beginning. So far, the data doesn't have shown any artifacts. I mean, of course, we tested this in advance. The only big artifact that we saw at some point, it was when we were using the rubber buffers for the short channels that had metal pieces in them. But apart from that, so far there is nothing that indicates, that there are any artifacts in the signal. Of course, now we are publishing the preprint and finalizing some of the analysis, but everything so far is looking, that the data quality is comparable. Yeah.
Okay, awesome. Thank you so much, Sarah. We don't have any further questions from the pre-registration, but people, this is your moment to ask any last burning questions that you have. There's this amazing preprint coming up, but here you can ask the questions face to face. So,
and if anybody is thinking about doing implementing this type of a study, please do contact me, because we've gone through so much troubleshooting that I'd be happy to share the expertise.
that is super kind of you. Thank you, Sara. So I think then in the interest of no further questions, oh, sorry, I thought that was a question. Somebody was just thanking you. So thank you so much, Sara, for your time, for your amazing work. I'm looking forward to seeing the publication out. And thank you everyone for attending the webinar. As I said before, it's recorded, so it's all available. And we'll see you in the next one. Thank you.
Thank you.